Hello, everyone. Welcome to the latest presentation of the Rift Valley Network webinar series. My name is Federico Falletti, and I am the host for today's talk. If you're participating in the live webinar, you can submit questions or comments in the chat at any time during the presentation, or you can ask voice questions by raising your hand once the presentation is complete. Today's talk is by Dr. Matthew Knisley. Matthew is a postdoctoral research scholar in the Department of Anthropology at Washington University in St. Louis and a research associate at the Missouri Botanical Garden. His research explores the history of forager landscapes in Eastern Africa, drawing on historical archaeology, political ecology, and science and technology studies. He's also interested in the Anthropocene, industrial food systems, and invasive species. Please let's welcome Matthew Knisley.
Thanks for having me, everybody. I always enjoy being able to present to this group, and the conversations with you are always great fun. So I will get started. And the general outline of how I'm going to give this talk is explain the comparative archaeobotanical collection that I have been working with my friends and colleagues in Tanzania and elsewhere to build since 2023. I am going to walk through sort of how and why we settled on this project, how we are actually accomplishing it. And then I'll give you some brief examples of how we are putting it to use. And what I have also done at the end is listed some issues that we could talk about as a group to think about how the work that we're doing could be of benefit to the folks in the Rift Valley network. And if you were interested in doing this kind of research, some of the kinds of funding and other logistical issues that you could think about. And also how you could potentially incorporate the collection that we are building into your own research. And as I'll explain, this collection, part of the goal behind it was to make it publicly accessible over the long term. And so we've partnered with institutions, not just in Tanzania, but also in Kenya and Ethiopia and the United States in order to expand access to this collection. So as a bit of just basic overview, I thought I would walk you through what archaeobotany and paleobotany are. These are archaeobotany is a sub-discipline. It's really a set of research techniques within archaeology. And archaeology, at least in the United States, institutionally is often a sub-discipline of anthropology. Paleobotany is related, but is often more aligned with botanical and geological research. And I'll walk through some of the difference there. But most simply, archaeobotany is the study of ancient plant use, and paleobotany is thinking about the history of plants, and so is oftentimes taking a more evolutionary kind of focus. And I'll walk through some examples of that. But archaeobotany, at its most basic, is the study of ancient plant use, and there are many topics that archaeobotanists explore, including domestication and spread of domestic plants, things like past diet and changing diet as people begin changing economic strategies from foraging to farming to pastoralism and moving back and forth between those things. And then also issues like anthropogenic impacts on the environment. This kind of research is really has a long history. It has been done in some form or another for about a century. It really started to emerge as a field of its own, a set of techniques in the 1960s and 1970s as archaeologists and anthropologists became much more interested in issues of environmental adaptation and thinking about how culture and especially material culture reflected adaptation. That's not necessarily the theoretical framework that we use now, but that was the context in which a lot of the foundational research emerged. And as a form of research, it's really well established in certain parts of the world and has been done very little in other regions. And there's many institutional reasons for that. Some of the places where this form of research has gone on for a very long time would be North America, uh, the Middle East, Western Asia, Southwest Asia, and Europe to a certain extent. Also Central America to a certain extent, South America. It really hasn't been done as much in Africa. And a lot of the work that has been done in Africa has been focused on early farming. And it's also focused on, and as a result, is focused on domesticates. And so there have been instances where wild species have been recovered, but without a comparative collection, unless the person doing the research is familiar with the flora of a particular region, the wild species remain unidentified. And so much of the research is focused on the emergence of domestic plants in Africa and has been somewhat geographically restricted. So in East Africa, most of the ethnobotanical work has occurred on the coast in the context of Swahili coast archaeology, urban archaeology. And then we also have a lot that's been done in West Africa. And to a certain extent, there's been some work done in the Great Lakes region. But in most contexts, focused on domestication, diet, and to a certain extent, trade. Paleobotany, there is a long history of doing paleobotanical work in Eastern Africa. That research focuses more, because it's especially dealing with longer timescales. So actually, let me pause and back up. There is also a difference in the kinds of evidence that some of these different fields focus on. So in archaeobotany, there are several ways that plants can preserve. One is if you have water and especially low oxygen environments or deserts, you can sometimes get good plant preservation through waterlogging or desiccation. And in Africa, in many places in Africa, you actually have, because of the wet and dry seasons, you have too much humidity. And so organic plant remains in most contexts are going to decay. So what we typically find in archaeological context, there's several sets of material. One is carbonized material. So things that fall into fire or burn, either on purpose or on accident. Most of that will just be destroyed by the fire. But a small portion of it the carbon will remain and it will basically turn into a piece of charcoal that looks in large measure, and I'll show you some pictures later, like it did before it burned. And so you can find, and what we often find are things like seeds and dense plant parts like nutshell, things like that. So the harder parts. Things like leaves tend to burn. We don't find those carbonized, although you can sometimes find things like grass stems. And with other plant parts, and so the other issue is, to a certain extent, the way the food is being processed and consumed also matters. So if you're eating something like leafy vegetables and you're boiling them, those are not the kinds of things that we're going to tend to find. But if you are cooking grain, especially if you're cooking whole grains, if some of those fall into the fire, we might find those. Roots and tubers, many times those are pounded, and so they're also unlikely to carbonize. But there are other ways that we can approach it. So in recent years, people have started to develop methods to look at things like lipids that soak into ceramics during cooking. And so you can infer whether plants or animals, and to a certain extent, even particular kinds of species that are being cooked in vessels. If you don't have plant parts that carbonize and can be recovered, you can have, there are other, and in archaeobotany, we'll often refer to things like seeds as macro remains. And so those are things that are visible with the naked eye. Although in order to actually identify them, you'll need to look under a microscope. Then there's another category of remains called micro botanical remains, which are things like phytoliths, which are silicate structures that help with various things in plants, but often form in between cells in plants. And when the rest of the plant goes away, those little silicate structures remain and you can recover those. And some of those you can actually identify sometimes you can only identify them to fairly large groupings of plant families. Sometimes you can actually get them down into genus and species because they're morphologically distinctive. And then we also have things like starch grains. And so if you have something like a root or a tuber that has been processed on a grindstone, and I'll come back to that, and you can actually see an example of that in the present here on the slide, you can actually recover those kinds of materials from grindstones that you recover archaeologically. And similar to phytoliths, in certain cases, those starch grains can be identified to fairly large sorts of groups of plants, but in other cases, you can get them down to genus and species. And when we're doing archaeobotanical work, our goal is always to get it as close to species as possible, because that allows us to be a bit more specific in the kinds of questions that we can ask of the record in terms of the particular plants that are being used and how they're used. Paleobotany looks at things like phytoliths, but also pollen. And part of that is because phytolith and pollen, due to their structure and what they're made of, are more likely to preserve over a very long term. And so you can study plants and use those to reconstruct climate and things like that over a much longer time period than what archaeologists are typically looking at. So Africa, there is a pollen database that's been around for quite some time for Africa. That's been used to study in many studies of human evolution and also, as I mentioned earlier, paleoclimatic reconstruction. Similar techniques, similar questions, looking at similar kinds of remains. But archaeobotany, and especially this project, is really focused on what I was describing as the micro and macro botanical remains, so seeds, roots, tubers. And we were also trying to collect samples so that people, some of our colleagues and future researchers, can obtain especially phytoliths from the plants that we've been collecting, although that's not a particular focus of our immediate research. And I'll go into some of that to make it make more sense, but that's a very high level overview. One of the challenges with trying to do archaeobotany in general, but also especially in Africa, is that there's relatively few people who are trained in it. The fieldwork portion is relatively straightforward. So if you are working in a context where the plant remains that you are recovering are typically carbonized, the method that we use to recover those remains is called flotation. And during excavation at an archaeological site, there's different ways of doing it systematically, but you would take sediment samples and then you place those sediment samples in water. The dirt, the sand and rocks and everything else are heavy, and so they will sink to the bottom. But the carbonized material will actually float to the surface, and you skim that off, you dry it, and then you can take it into the lab and begin identifying it, sort of doing a miniature excavation again to identify plant remains in the material that floats up. So the fieldwork component is relatively straightforward. We know we have for a long time have known sort of best practices for recovering this material. There are all sorts of complications that get in the way, but that one of the biggest is access to water. But as long as you have access to water during fieldwork, it's relatively easy to recover this kind of material. The problem is that the lab work really takes a long time, and you need a comparative collection in order to identify the plants that you're finding. In Africa, for a long time, folks thought that something about the soil chemistry was not conducive to the long-term preservation of carbonized plant material. It turns out that might have been an absence of evidence rather than evidence of absence. And so some people had tried at a few archaeological sites, didn't have much luck. That does happen sometimes, that things just don't preserve. And partly for that reason, and also because people just simply hadn't been trained in it, many people just didn't even bother to try to recover the material. But as people have actually gone to archaeological sites and really focused on trying to collect this material, it turns out that at many sites it's there and it's abundant and it's perfectly good material. Limited plant preservation can be an issue, but at this point, especially in understudied contexts in Africa, that's something that we should really establish rather than assert that's the case. So we should at least try. And then, as I mentioned, a lack of comparative collections means that even if you find the plant remains, you might not be able to identify what they are. So there's a whole range of factors to take into account here. Really, for over a decade now, various friends and colleagues of mine who do archaeology in Eastern Africa have lamented the fact that there are relatively few of us who have done archaeobotany, especially because this region of Africa is so important for understanding many aspects, not just of human evolution, but later human history in terms of thinking about changing economies related to especially the spread of domestic plants and animals. Eastern and Northeastern Africa are thought to be based on various lines of botanical and genetic evidence where sorghum and millet were domesticated, which are major global crops, and also spread throughout Africa and Asia really very quickly and very early. And Northern Africa is thought to be one place in which cattle were domesticated, and East Africa was one region through which domestic livestock spread to the rest of the continent, including goats and sheep, which came in from Asia. So Eastern Africa is a really interesting region, and there's been a lot of zooarchaeological work, which is the study of animal remains. But there'd been really, I mean, effectively no archaeobotanical work. So we know that this region is interesting in sort of in thinking about it in sort of a global comparative lens. But there was this huge body of evidence that we just simply lacked, partly for all of the reasons that I've just walked you through. So for years, we had been lamenting this fact, and we kept saying, we should really do something about it. And there have been some really good efforts. So our colleague Sarah Walshaw had started building a comparative collection focusing mainly on domesticates when she was, she continues to work on the coast of Tanzania. Uh, and and we wanted to sort of follow her example and build something that would be a bit larger and more focused on wild species. And one of the things that we wanted to do as we were talking about this is, well, if we were going to do it, let's think about these collections that exist in other parts of the world and think about some of the problems with them so that we can design our collection to try to prevent those from being a problem in this context. And a few years ago, my colleagues Mary Prendergast and Kate Grillo, who I believe have spoken to the group before, they've been working at a site in Tanzania called Luxmanda for about 15 years now. And they were planning to apply for a grant to sort of wrap up their work at that site. They had a few large questions that they still wanted to answer, but then they were interested in sort of winding down their work there and continuing on new projects. So they applied for several grants. It turns out they were quite lucky and got them all. They were only expecting to get one. And they then approached the funding agencies and said, as part of those grant applications, they had proposed doing a small scale comparative collection that would help us do archaeobotanical work specifically at Luxmanda. And they went to the funding agencies and said, could we reconfigure the funding slightly since we got all of these grants to bring me on board to really focus on building the collection and thinking about how we could, rather than having that be mainly focused on Luxmanda, how we could approach it to where we viewed it as more of a pilot project for future work. And so I got brought onto that project in 2022 to 23. And then in 2023, we applied for additional funding from the NSF. We presented our initial results from the work at Luxmanda and said we would like to expand the scope of this. And so that's what I've been doing since 2024, using this NSF funding to expand the project from Manyara, near the Luxmanda site where we've been working, into the Sandawe homeland and also southwest Kenya. So extant comparative collections face several problems. One is when in the 1960s and 1970s, many of the early archaeobotanists were trained in botany. And so they were familiar with plant taxonomy, with collecting plants, voucher specimens on the landscape. That's become increasingly not the case. So archaeobotanists are trained primarily as archaeologists, not botanists. And there are very few botanists who are also getting archaeological training. So we have comparative collections that have been compiled by archaeologists where the identifications may have, the botanical taxonomic identifications may never have been correct, or if they were correct at the time, have not been changed to keep up with changing plant taxonomy within botany. Another issue is that is curation. So many of these collections are housed at universities in anthropology departments and often in the labs of the specific researchers. And what we have seen is that there are instances where these collections, upon retirement or funding and education priorities at universities, where those collections will actually be discarded. And so they take a huge amount of time and money to put together. And they're absolutely critical for doing this research. But if they get tossed, all of that work is gone. There's also an issue that if a particular professor is really busy or is simply feeling unhelpful, that there's a potential that they could be a gatekeeper that's preventing access. So if it's in an individual's lab versus a more publicly accessible place, access can be limited. And now, in recent years, what we're also seeing with changing politics is many of these collections are stored in North America and Europe. And so if we're thinking about archaeobotany in Africa, other than some limited cases like Sarah Walshaw's collection, like I mentioned, that's stored at UDSM, researchers would then need to also have the funding, but also get visas to go to the countries where these collections are stored. So then there are international limitations on access to these collections. So as we were trying to keep all of this in mind as we were getting started on this research, and we put together 5 principles that we wanted to focus on. So one is we wanted to strive for comprehensiveness because these are so time consuming and costly. We wanted to make sure that we were doing as much as we could with the limited time and resources, not just to facilitate our own specific research questions, which I'll talk about later, but also to provide resources that would be useful to others, perhaps even in ways that we can't anticipate right now. So one of the things that we were doing before we started the field work for this project was we did a literature review in all of the different regions where we're working to try to understand what are all of the species that have been recorded through whatever means that have known uses for food, medicine, animal fodder, building material, whatever. We also wanted to do some new ethnobotanical research to try to understand contemporary plant use. And then we also wanted to gather a wide variety of specimens that would make this collection maximally useful to anybody who has reason to use it. So in addition to collecting botanical voucher specimens, which especially focus on things like flowers, we wanted to gather the materials that would be helpful for archaeobotanists. So we wanted things like seeds, shells if a plant produces hard parts, roots and tubers, leaves, extra leaves beyond what just goes on the voucher specimen that could be used both to sort of compare what we're finding at archaeological sites to the collection, but also in the case of, so we wanted to also collect extra material for things like destructive sampling. So in order to study phytoliths and pollen and things like that, you do need to destroy using acid or carbonization. There's different techniques in each case. Different parts of the plant in order to obtain those very small structures like the phytoliths. Excuse me. In the case of seeds, it is sometimes when seeds carbonize, they don't change in shape. Sometimes they do. And so it's sometimes necessary to take modern seeds and to carbonize them so that you can understand how their morphology changes when they're exposed to heat. So in addition to having the samples, we needed to have lots of different kinds of plant parts that are not necessarily or normally typically collected by botanists. But we needed enough material that some of it could be destroyed in order to do some of the kinds of research that we're talking about. So we wanted a wide range of species and a wide range of samples of each of those species to be collected for the collection. So we wanted to be as comprehensive as possible. And also because every time you go back, that of course takes more time and more money. So we wanted to sort of do as much as we could when we could with the money that we had. As I mentioned, we wanted to rely on botanical expertise. My friends and I and colleagues, many of us had experience doing small scale botanical collection, but we are really not trained in it. We are not professionals. And so we wanted to rely on botanists who are much more familiar with the landscape and know how to produce high quality samples for long-term curation efficiently. And so we learned a lot from them doing that. We wanted to curate an herbaria. So herbaria are basically plant libraries is one way to think of it. Leaves and flowers of a plant will be slowly dried, mounted on a piece of archival paper, and then organized and curated over the long term and available to researchers. The National Herbarium of Tanzania is in Arusha. University of Dar es Salaam also has an herbarium. We are also partnering with the East Africa Herbarium which is associated with the National Museum of Kenya in Nairobi, and the National Herbarium of Ethiopia, which is associated with the University of Addis Ababa. And so we also wanted to prioritize institutions that are open to the public for research. In order to ensure accessibility, we wanted to collect. So I mentioned that we tried to collect many kinds of samples and as many species as possible. Each one of those species and each one of those samples, we collected 6 duplicates of in order to distribute to the 4 different herbaria in East Africa that we're working with. 2 copies of them come to the Missouri Botanical Garden in St. Louis. The Missouri Botanical Garden has one of the largest herbaria in the world, and they will curate one of the sets long term, and then the 6th set is available for specialist analysis or exchange with other research institutions. So herbaria will often exchange material back and forth in order to help each other's research programs. So we also have this sort of extra set. So we aimed to make it geographically dispersed, but we're also digitizing it so that folks who can't get to those physical locations can also see high quality images of the plants and the associated tissues online. And on this slide, you can see a seed that we photographed, and this is called, the genus is Dactylotinium, and it is a small grass seed that is consumed in much of East Africa, but it's very small and very hard to process. And so it's typically reported as something that people only eat during times of food stress. And then we also wanted to publicize how we were going about the project so that others could recreate what we're doing. And basically, our hope is that we can begin building smaller collections that are focused on specific sorts of research questions, but that we have sort of a unified digital presence that people could contribute to following sort of standard procedures. So we were also thinking about like, we're doing this now for our particular research projects, but if folks wanted to take this and do it in the future, we need to make sure that they know how to do it so that they can replicate it. When we first started this work, it was in the context of this archaeological project at Luxmanda. And Luxmanda, which has been discussed here in this group during previous presentations, is a really interesting early pastoral site in Tanzania. It is the earliest evidence of domestic livestock in that part of the world. Contemporaneous sites are much further to the north. Luxmanda is also very interesting because it's much larger than many other early pastoral sites in Kenya. And it also, interestingly, has lots of grindstones. And it has long been speculated, partly because we suspect that sorghums and millets were domesticated in Eastern or Northeastern Africa, that early pastoralists may have been involved with the domestication of grains as well. But there's no good evidence because nobody's really bothered to look. And so we wanted to do some ethnobotanical work to understand how do agropastoral communities living near the Luxmanda site interact with wild plants now? Which kinds of species do they use? How do they use them? So that we could begin gathering plants that might help us identify not just seeds, but also starch, grains, and phytoliths that we were recovering from the archaeological sediments and the surface of those grindstones at Luxmanda. And we understand that climate has changed quite considerably in recent millennia in Africa. And we also know that contemporary lifeways are not going to be representative or identical to what was going on when the site at Luxmanda was being created, but it's a good first start. And if it turns out that the collection doesn't help us understand these plant remains at all, although spoiler alert, it has helped us, then you go from there. But it's a good first start to just sort of start with what's on the landscape near a particular site. And so we did, in addition to the ethnobotanical literature review, we did one-on-one and group interviews with folks about species with known uses. We also did focus groups to develop community calendars to understand how plant use changes across the landscape and over the course of the year. We did what are called field walks, which are when you just go out on the landscape and talk about useful plants with folks in a community who are understood to have a lot of that kind of knowledge. And then sort of simultaneously to that, we had various teams of botanists and others collecting specimens of all of the plants that were being mentioned. And like I mentioned earlier, everything we were striving to collect 6 duplicates of. Duplicate is the word that's used in botany, even though it's more than 2. So we were trying to collect 6 sets of all of this material. Then when we got back to the herbarium in Arusha, the botanists were confirming the identifications that they had assigned to these specimens in the field. We then we're still in the process of getting everything to all of the institutions internationally. And we are also currently in the process of digitizing, making high quality photographs of all this material. And as I'll talk about in just a few minutes, we are using it to begin exploring some specific research questions at Luxmanda, but also an archaeological site in the Sindawe homeland and an archaeological site in Southwest Kenya. So I'll walk through some pictures really quickly of the research team in action. And these were just snapshots that I took from doing the field work. So in 2024, we were working in Manyara. 2024 and 2025, we were working mainly near High Dome. In 2026, the work has focused in the Usandawe homeland in Chemba district, which is in Dodoma region, so a little further to the southeast. And this picture on the left is an example of we've collected plants in the field, and we're in the process of putting them in newspapers. And then once they're in the newspaper, we put them on drying racks that are over a heat source for several days until they're completely dry. Then once we actually get to the herbarium, the material are further preserved by being put in a freezer for several days in order to kill any insects that would be on the plants. Some other pictures from the field. This was our botanical team. Yahya Abeid, Charles Msagwa here in the middle, and Moses Mwangoka. And collectively, they have decades of experience doing botanical work all across Tanzania. And it was really great because they were teaching us how to do botanical work, but then we also got to teach them a bit about the specific questions we have and how we're interacting with the plants in ways that botanists don't typically. And as I mentioned, we're really interested on getting the seeds and getting lots of the seeds, which required extra work to figure out how do we actually dry these things quickly so that they don't rot or get other kinds of mold and mildew on them. And so here on the right was when we were working with a local carpenter to build special drying racks for all of the extra seeds that we were collecting. We were also doing this bulk collection of things like leaves, which you need for the microbotanical analysis. So I mentioned in order to obtain the phytoliths and pollen and things like that, you actually have to destroy certain parts of the plants. And so we were collecting extra material so that the botanical voucher specimen sort of remains untouched and housed in these herbaria, but we have these associated extra tissues that researchers, if they're interested in particular species, can take for the destructive sampling. And another thing that came up, sort of a side quest as we were doing this, many of you may know Herman here in the picture. We became really interested in geographic place names, and he was very helpful for transcribing. So we were trying to write down local names for plants. We also became interested in looking at some of the historic topographic maps and thinking about the meanings of place names on the landscape. And so that was a side project we hope to return to. But some high-level results so far is that across 2 seasons of fieldwork in Manyara, we've identified over 1,100 species with known uses. Of those, we have almost 800 where we have collected botanical vouchers for, which are the pressed leaves and flowers that will be curated in the herbaria. And about 642 of those we have been able to collect seeds of. In 2026, just in February and March, we were doing some collecting in the Sandawe homeland. There we've identified over 600 species with known uses. 376 of those have voucher specimens, and 150 to 200 we're actually still figuring this out because we're compiling notes and things like that. We have the seeds. And the reason you can't do them all at once is it's very time consuming. Not every plant is easy to find on the landscape, but also things are in flower and in leaf and producing fruit at different times of the year. So doing this research will often entail needing to go back multiple times to collect samples at different points in time. Since then, we have also built infrastructure for a macrobotanical laboratory at the National Herbarium of Tanzania in Arusha. There is already such a laboratory at the University of Dar es Salaam that Sarah Walshaw helped build. We're in the process of creating the digital collection, and I'll be able to share that with you soon, but we're still in a testing version, so it's not quite ready to be live. And we are also using the remains to begin answering specific questions we have about Luxmanda and Marabou, 2 archaeological sites in Tanzania, and also this archaeological site of Kakapel in Kenya. And and here are some pictures of some of the seed collections. And some of our work at NHT and in St. Louis of trying, there's just a lot of logistics to manage this much material. And so the picture on the left is us sort of sorting everything to distribute across the world in Tanzania. And this is a package of some of the material that had arrived in St. Louis. I also wanted to have a picture of our team because it is truly international in scope. We have folks from Tanzania, Germany, Spain, the United States, all working on this and addressing different aspects of the project. Really quickly at Luxmanda, the thing that we're trying to do, a lot of work, both archaeologically and ethnographically on pastoral communities, has really focused on the milk, blood, and meat triad. But a lot of the ethnographic work demonstrates that pastoral communities rely very heavily on a lot of wild plants, and we've really not been able to explore that archaeologically. As I mentioned, we've recovered seeds and starch grains and phytoliths from the sediments and also those grindstones at Luxmanda. There's some really exciting results that we hope to be able to share with folks soon, but it's still a little too early. And I'm not, we just need to do some confirmation of what we think is going on before we start telling folks about it. But really exciting and really looking forward to being able to share those results with people. At Cockapel is this very interesting site with and so Luxmanda is about 3,000 years old. Cockapel has a archaeological sequence that runs for about 15,000 years. The lower levels, the older levels, have evidence of foraging. There is some evidence of domestic livestock, and then the upper, more recent levels do have evidence of agriculture. And in fact, so this was before I came on board. This was my friend Natalie, who's a professor at WashU, had done some archaeobotanical work there, and they found some of the earliest evidence of domestic plants in Kenya from this site. And what I've been doing as part of this sort of the expanded version of this project is I am sorting and analyzing the wild species from those lower, older foraging levels at the site. And we're also finding lots of interesting things there. It'll take a little longer to figure out what's going on with that, just because we're just now getting to the process of trying to use this collection I've just told you about to figure out what it is that we have found in those samples. And we also have plant remains from the Marabou rock shelter, which is from the Sandawe homeland. And that archaeological site is fascinating. That sequence goes back at least 50,000 years to recent centuries. I have really not done much analysis from that site yet. We do know that it does have plant remains in the sediment samples that I've collected. But one of the interesting things about that site is it has some really old ostrich eggshell beads, which are interesting to archaeologists for all sorts of reasons that I don't need to go into now. But as I've previously told you at this group, my research in that, so the Sandawe have been thought of as being this sort of descendant foraging community. I did an archaeological project to try to get a better sense of what kinds of evidence we have for food production, so farming and pastoralism, but also exchange with other places in that region. And the archaeological record is really presenting a much more kind of complicated picture than what we had thought about the history of that community and that region based on things like oral history and ethnography. And so I'm hopeful that this archaeological site and by using the collection, we can begin to understand sort of the long-term history of plant use to further fill out that picture that has been emerging from my earlier archaeological work. So those are the places where we're using this project, but it's open to other researchers. We're hoping that other projects will do sort of smaller scale botanical collections in order to expand the geographic scope. And the collection is available and can be used in all sorts of ways other than the specific questions that we're bringing to it. And we're hoping that moving forward, that we can begin training students. So all of the universities not all of the universities, but Tanzania, Kenya, and Ethiopia, students now can do graduate training, undergraduate and graduate training in archaeology. And we're hoping that we can begin identifying students who are interested in this kind of research to begin training them using this collection so that we can begin expanding the field of archaeobotany in Africa beyond this. And where I would like to leave it is on this slide is some things to think about and discuss. So one is costs. On one hand, like, yes, it is expensive, but it's also not that expensive when you think about sort of the per sample cost, given that we've collected, we will have collected well over thousands by the time this project is done. So on one hand, yes, it costs a lot. On the other hand, it doesn't cost as much as you might think. One of the issues that came up that I haven't really talked about, I didn't have quite have time to talk about, is how this work, some of the new ethnobotanical work we did, but other kinds of aspects of this project could help facilitate interdisciplinary research and especially linguistic research. So we were lucky in Manyara to have worked with Herman, who was able to help us transcribe the Datoga plant names. We were also able to use various dictionaries and things like that to try to begin transcribing some of the Iraq names. But one of the challenges we had is we're not trained as linguists. So we're trying to collect this information. We're trying to transcribe names to the best of our ability. We don't have this particular skill set, but maybe moving forward, one thing that we should think about would be more targeted kinds of recordings or interviews to get examples of the names being said. Another way to think about it is these materials are now being housed at the National Herbarium. So linguists could go there with their interlocutors and actually show them the plants. Now, a dried plant specimen isn't going to look like the plant on the landscape, but many times it is still going to be identifiable and recognizable. So there are ways that I could imagine linguists using this collection. There are lots of interesting ethical issues that have come up. So in the North American context, just as one example, for many decades, researchers have been sometimes voluntarily, sometimes really against their will, unfortunately, have been rethinking the nature of how academic researchers have engaged with Native American communities. And many of those groups, but not all, now have procedures in place for obtaining community consent for particular forms of research. In Tanzania and in Kenya, there are very different kinds of research and permitting processes. But one of the things that we've talked a lot about is, we went through the appropriate steps to get research clearance, but some of these kinds of plants might be sensitive information related to, if you could imagine a plant that might be used in a ritual context. And so I think that there's room for us to think a little bit more about, is all of the information that we've been collecting about these plants really appropriate to be sharing in these public venues as we've designed it? How could we better think through getting community consent on research sort of beyond the formal legal process of getting research permits? So something to think about. And then there are limitations of the data. So partly due to time constraints, we were operating on the presumption that each sort of botanical scientific species, has a local name. That's not how things work in practice, as many of you know. Sometimes a name will apply to many species and vice versa. Sometimes a species will have many names. So this data set, the way that it was constructed, we simply didn't have the time to really go into some of those kinds of nuances. Those are the kinds of things that, like, many of us here might immediately recognize as being a data limitation. But I also do think that the onus is on us to in our publications to sort of to mention this as a possibility, to think about as a way to improve the information we've collected, but also so that people don't make the wrong kinds of assumptions about what's being represented in this collection. So I'll leave it there if you'd like to ask questions um, about the presentation or some of these other issues about how to do this kinds of research or how we might be able to work with linguists moving forward. I'd be happy to have a chat.
Thank you so much, Matthew. That was super interesting. It's nice to learn about your work. I never heard anything about it, and yeah, it's really, really cool.
Thank you.
If anyone has questions, you can raise your hand or you can write something in the chat module, and I will read it out. Martin?
Yes, thank you, Matthew. Very inspiring. And also in the last slide, invitation for collaboration. One of the points that you raised about the problems with species, name, etc., very true, and is also a problem. Although I'm aware of it as a linguist, the dictionary, Iraku dictionary, if you used it, is not perfect in this respect at all. It's true that when I tried to give the scientific names to plants that I knew the Iraku name for, it was very helpful to use all those collections by Tanzanian botanists, where maybe the rendering in Iraku was far from perfect, but still good enough for me to recognize what was meant. So I think it is it we don't have to do the other discipline perfectly in order to cooperate. And I think even it's probably more valuable if we cooperate and we don't learn all each other's disciplines. So I in ways in sharing data that would be great. I think I should send you some of the databases that we collected for names for the area. And yeah, I had great plans with somebody from a completely different discipline in sharing each other's materials from socialization, the children's socialization, uh, using the interviews that I would do interviews that she would then be able to use in analyzing in content, and that I would be able to use analyzing linguistically. So I would be very interested in your interviews. I think those things among our disciplines are probably much more easier than the ones with the children, because of the ethics, because they had all these problems with ethics. That was the ethics permission was given for one specific goal, and this is potential problems sometimes in sharing each other's collected materials, which I think really is a shame, because it's not only more efficient to use each other's materials, but also you learn so much from it.
Yeah, and one thing that I didn't go into this, but there are, so a lot of this information will be accessible in one form or another. So the digital database that we are constructing, my friend and colleague Natalie are working on, is the database of photographs of seeds. But and so that the first version of that is going to be relatively simple, where it will be the species and then the photographs of the seeds and some basic descriptions of the seed, so size, shape, texture. But we are also, that site, each species will also be linked to the Tropicos database that is housed at was developed and is housed at the Missouri Botanical Garden. And that database has images of the voucher specimens that we collected, but it will also have a brief summary of the field notes that we made about the species. And so that will include the uses that were reported to us, but it will also include, when available, the names of the species in Datoga, Iraq, and Sandawe. So it might be a little clunky to use. We're still figuring out how, like, what the mechanics of all of this will be. But this is the sort of thing that if any of you are interested in, we would be able to do a batch download of this data to share with you. And what we were also doing, and this is, I didn't mention this because this has come up just within the last week or so, but we are going to use this project to help the Missouri Botanical Garden test out some new functionality, relatively new functionality that they've built into their database for ethnobotanical databasing. And so what we're hoping, it's not, I can't promise that this is going to happen, but ideally what we'll be able to do is have a portal that will be available where you can search about known uses of species, and that will draw upon like our research that we did, the voucher specimens, and the literature that we've compiled. And so we're hoping to bring that all together in one place. What exactly that will look like and the extent of its functionality is unclear just yet. But all that to say, we are hopeful that that all of the information that we've been collected will be available and searchable and usable in some way. But in the meantime, if there are specific things that you would be interested in, like the word lists, we'd be happy to share that.
Yeah, thank you. Yeah.
Yes.
Bonnie, do you want to ask a question?
Yeah, questions. Oh, good. Oh, I have three. So my first thought was, yeah, when you're trying to get your 6 specimens, I immediately thought that's a problem if you're going by the name. So did you tend to get them from the exact same location, all 6?
Yes. So oftentimes it sort of varies from species to species. So we tended to do it in the same location. But this also does raise an issue of there are some species where the flowers and the fruit are never on the plant at the same time. So some of this, and so there are ways that that botanists in the field can mark particular specimens or particular collection areas to be able to return to them to try to make sure that you're actually getting material from sort of the same population of plants. But yes, this was definitely something that we considered, but it's realistically, you can't build a collection like this. I mean, you can get a good start during one field season, but to really be comprehensive and complete, you've got to do repetitive field work. But then, yes, you are correct. This brings up the issue of uh, gathering from the same location or the same population, and then that there are also potentially that's making a lot of assumptions about whether you're actually doing that, but also the extent to which sort of local taxonomies map onto the botanical taxonomies.
And then are there a lot of notes about the geology of where these things grow?
Yes. Yeah, we do try to take those notes. And one thing that came up during the interviews was that there were several plants where people that were recurringly being mentioned, but folks were saying, but they don't grow here. You'd have to go to this other region. And so we did set aside time to we did a little bit of research in the No Forest Reserve, NOU, which is it's north of High Dome near Mbulu. And we also went down into the Yaida Valley to do some collection because there were certain species and certain kinds of species that didn't grow where we were working.
Yeah. Punchupe, for instance, I had to take a trip to get to that one. But yeah, well, this is such a huge project, and the logistics just seem like a nightmare to me, like mailing plants or taking them on a plane. I mean, it must have been pretty nerve-wracking to make sure these
Oh
things.
yeah, it has not been easy. The very first shipment made it all the way to Chicago on its way to St. Louis, and then got sent back because we hadn't caught that there was an error in the zip code. But everybody understood. Uh, we spoke with customs. They're like, we understand this is important material. We're not going to destroy it. We will send it back. You can fix the paperwork, send it back. So we did. Then last summer, we had a shipment that was, I believe, had made it to, oh gosh, had it made it to Ethiopia? Well, so before it even left, when we paid for the package, everything was fine. But then KLM, it was going to go with KLM freight. They had changed their weight limits on packages. And so they would no longer take the package because they were like 2 kilograms overweight for their new policy. But they switched to Ethiopian, which had a slightly higher weight limit. The packages then made it to Addis, I believe, if I'm remembering correctly. And that is when, due to the diplomatic disagreement over tariffs between the US and Tanzania, reached a peak, and packages were no longer being allowed to be sent. And so it got returned to Tanzania. So yes, we have, and we're actually still, and so we have a material at NHT. Everything is, we also have all the material that's going to UDSM in Dar es Salaam. That's done. We almost, actually next week, are hoping that all of the remaining plant collections that we've done to this point will be en route to the US again. But then we still have to get the material to Kenya and to Ethiopia. And in all of these cases, like each country has a different set of permits. And our collaborators who are doing the starch and phytolith analysis are in Spain. So we've also been sending material to Spain. So each one of these countries has a different sort of permitting regime that you need to follow. So yeah, it is very logistically complicated, but we've been able to manage. Yeah. And it's also, you're right, just the day-to-day logistics. But I'd also be happy to talk to you about the logistics. Herman was an amazing collaborator. We ended up doing things like lunch boxes. We found people in High Dome who could make us lunch boxes. I believe maybe Andrew had given us that tip. Maybe it was Herman, I don't remember. So that we could stay out all day and do the field work. Yeah, it gets very logistically complicated, but it's fun.
Matthew, while you were like among your field notes, did you get any stuff where people were talking about hot plants versus cold plants?
No, but I'd be very interested in that.
This was something that came up just very briefly when Alice Mitchell and Crispina Alphonse were doing a workshop on ethno-linguistics, and they came to High Dome and spent sort of a day in the field doing plant identification. And one of the things that the people who were taking us on these walks, one of the big divisions that they were making was they would say this plant is hot and this plant is cold. And so hot generally corresponds to plants that are thorny and cannot be used in traditional medicine, whereas the cool plants are the ones that are not thorny and can be used in traditional medicine. What's interesting about this is, it seems to be pretty widespread. So outside of, say, for example, the Datoga community and the Iraq community, Todd Sanders writes about it in his dissertation. And I've asked a few Tanzanian colleagues about this, and they say the same thing. Yeah, there's hot and cold plants, and there are certain things that you can do with the hot ones, and certain things that you can do with the cold ones. I just find this interesting. I was wondering if you came.
Yeah. No, we didn't come across that, and some of that is because we had some very specific questions that we were asking about. One of the things that we did notice, and this was on the part of the researchers, is so Kate Grillo has done ethnobotanical work with pastoral communities in Kenya. And I had done previous ethnobotanical work with the Sandawe. I talked about it here about leafy vegetables. And we even noticed it would be really amazing to listen to recordings of how we were doing our one-on-one interviews, because the number of fodder species that came out of interviews that Kate conducted is way higher than any of the fodder species I could record. But I recorded like dozens of leafy vegetable species, and Kate recorded almost none. So somehow we were both bringing our previous experience with the research, like maybe we had learned how to ask about these particular categories of plants, but there were real differences even among what we were recording and in some of the kinds of species that were getting listed. So no, we were much more targeted in the kinds of questioning we were doing, both on the one-on-one and group interviews and in the focus groups, really focused on particular uses that might help us with the plant remains we were finding at the archaeological site. So, I mean, that's another thing to think about with the limitation of the data is, yes, this is all very useful and provides insight into the use of plants in this region, but it should not be understood as providing like a comprehensive view of how of human plant relations, both in sort of like a economic but also a cultural perspective. But I mean, that's also a problem with any sort of ethnobotanical study. You know, many of them are going to be targeted on particular questions. But to answer your question, no, that didn't come up, but that sounds really interesting. Yes, Bonnie.
Yeah, some other question is, you want to know about medicinal uses, but I have a hard time eliciting medicinal terms because the categories of diseases are so specific and local, and I have no idea how to map them onto English words. I mean, that's probably not even an appropriate translation. So, yeah, it's like, do you have to sort of do some ethnomedicine before you can figure out the ethnobotany for the medicine, or and then it's like, will people mention things like treatment of STDs or abortifacients, and that's going to depend on the research. And yeah, with abortion being, it's illegal in Tanzania, right? So people, they might phrase it as, this is really bad, a pregnant woman can't do this, hint, 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 or something, but I don't even know if people, yeah, those are just the interesting issues for you.
Well, right. And you made reference to it as well, that gender of the researcher and the research participants could also impact the kinds of information that's being shared. And then most of our interviews were done in Swahili. And so there's also the issue of in the particular languages, like what kinds of translations are being made into the Swahili terms. So yes, certainly I think if folks were to, those are the kinds of issues I think that if folks were doing longer term ethnobotanical work, yeah, those are absolutely important issues to keep in mind. We did work with some translators in a few cases where folks felt more comfortable responding in Datoga or Iraq, but then it was translated into Swahili. So I'm sure that there's lots of information that is sort of mistranslated or lost along the way.
Since a lot of the medicinals might need to be boiled rather than, say, baked, in your experimental treatment of the grass seeds, well, I don't know that the grass seeds would be used, but yeah, could that make a different deformation of the plant remains? So, and one of the specific questions that we were asking about was, and again, this in this earlier ethnobotanical work that we were doing in Manyara, the ethnobotanical component was really targeted for particular research questions at the Luxmanda site, whereas the botanical work was trying to get those specimens, but also working out how do we actually do this in an efficient manner so that when we get funding to expand the project, we sort of have a sense of what to do. But for the ethnobotanical work, because of the grindstones at Luxmanda,
and because of the more recent abilities in doing residue analysis on ceramics, some of our questioning was focused specifically on sort of regardless of the use, what are the different plants that are processed on grindstones? And what are those plants and how are they used? And so then we wanted to make sure to get samples of those plants in order to either get, well, in this case, to get predominantly like the starch grains or the phytoliths to see if there were matches for what we were getting on the grindstones. And then similarly with the ceramics. So sort of irrespective of the particular uses, what are the kinds of plants that are typically processed in pots through boiling or whatever else? Yeah, so we were doing some of that kind of questioning as well that was really more focused on processing rather than our primary emphasis being like what specifically is it used for? Because we were trying to get a broad array of plants that we might be likely to find in the ceramics or on the grindstones.
Any more questions?
Thank you very much, everybody. And feel free to send me an email. And to your point, Martin, I have spoken with Andrew about trying to do some recordings of the plant lists. So hopefully we can move forward with that.
Yeah, thank
so
you.
if there is any way that you'd like to collaborate on anything like that, let me know. We'd be happy to put you in touch and to facilitate.
Thank you.
The work in Manyara is pretty much cleaned up at this point in terms of the data tables, the outputs. We're in the process of thinking about how do we actually make it accessible and usable. The Sandawe material, we need to do a little bit more data cleanup and processing, but certainly the Datoga we're ready to share.
Yeah, it'd be great to have it because there isn't much out there aside from what Bonnie collected a while ago and a few dictionaries, but not much on plants. Yeah. Great stuff. All right. Well,
thanks again, everybody.
Yeah, thank you so much, Matthew. And I will see everyone again. And I want to remind everyone that the next presentation is on the 29th of April. And it will be on Mbugwe Discourse by Lizzie Poole.